This is the last part of our evolution of populations video. So the study of classification is called taxonomy. It has been around for a long time. Scientists have always been interested in classifying new organisms that they discover and figuring out relationships and putting them into groups. There are different levels of classification. A single level of classification is called a taxon, or multiple groups would be called taxa. The person that is considered to be the father of taxonomy is Carolus Linnaeus. He created the system that we use today for classifying living things according to similarities. So our modern system that was created by Linnaeus has 7 taxa, and you may have learned this before. There's a little way to remember this. So starting with the most diverse, you would have kingdom. So for example, kingdom is going to include all animals. And then you go down phylum, class, order, family, genus, and species. Species being the least diverse because that's only one single organism like humans. And there's different ways to remember this. The one that your book has right now is King Philip came over for good spaghetti. I've also heard King Philip came over for grape soda. And there's various ones, and you might have been taught another one. So if you already know this with another one, then don't bother to learn about King Philip. But you will need to be able to remember what order these taxa go in. So a sample question that you might come across, it says the chart shows the classification of a dog and a gorilla. What is the lowest taxonomic level they have in common? So the way you're going to do something like this, you just go, they're both animals. They both are in the phylum Chordata, which means that they have a backbone, spinal cord. You have both mammals, and then you get down here where they have different order. Dogs are in the order Carnivora, gorilla is in the order Primates. So that would be your answer right there. And that's the type of question you might see for classification. So when scientists discover a new species, they're normally given the privilege of naming the species. A lot of scientists like to name the species after characteristics that it might have, or sometimes they name it after themselves. But each new species has a 2-part scientific name, okay, which the first name is the genus. The second part is the species. So here is Acer rubrum. This is the red maple. Okay, Acer is a maple, rubrum is red. I just happen to know that. You don't necessarily need to memorize all these scientific names. One thing you do need to know, though, is when you write a scientific name, the genus name is always capitalized and the species name is always lowercase. And they have to be either italicized, or if you're handwriting it, you can underline it, which does the same thing. It implies that it's supposed to be italicized. This system is called binomial nomenclature. Bi means 2. And this word basically means 2 names. And then nomenclature is a naming system. So a 2-name naming system describes exactly what it is. So Acer rubrum is one. Our scientific name as humans is Homo sapiens, which I'm sure you've heard before. But notice how it's written. The Homo is capitalized and the sapiens is lowercase, and it's in italics. So as we learn more about how things are related, we're trying to make sure that when we classify new organisms, they get put into the correct category based on what they're actually related to. In the past, they would put things together that just looked similar. So we've talked about how a shark and a dolphin look similar. Back in the day, they probably would have been put in the same category just because how they look. But today we know that they're not as closely related as they look. So when we classify stuff, we're looking at ancestral relationships, common ancestors, and how close that common ancestor is. So if they're together in one of the lower taxons, so like, for example, if they're in the same genus, they're going to be more closely related than if they're just in the same kingdom. So Also, for example, any two animals are going to be more closely related than a plant and an animal because they're in different kingdoms. So here's another sample question. It says, study the chart below. Which two trees are the most closely related? So again, you're going to start at the top. They're all trees, so they're all plants. And now we have, in the phylums, we have 3 that are magnolia phylum, and then we have 1 conifer. Remember, a conifer has cones. So we have to keep going. And then we've got 2 here that are in the same class. Okay, and those are going to be the most closely related. So that's the apple tree and the oak tree. So another example, we have the scientific name for red maple is Acer rubrum. There's a really pretty one right there. Would it be more closely related to the tree named Quercus rubrum or the tree Acer saccharum? Okay, and sometimes this confuses people because they see the rubrum and they think, well, that's the lowest level. They must be the most closely related. The name is 2 parts. Okay, so you have to look really at the genus to figure this out. So the 2 acers are going to be more closely related. This is the red maple, like I said. Acer saccharum is the sugar maple. And since those are both maple trees, they're going to be more closely related than Quercus rubrum. The reason this is called Quercus rubrum, this is an oak tree, but it's a red oak. So rubrum means red. So in this case, rubrum is just kind of a descriptive thing, and it's not indicating that they're more closely related. So look for that genus to see if they're closely related or not. So moving on from Linnaeus, we've added some stuff to our modern classification systems based on what we know about DNA and other ways that we can determine relationships. But first of all, let's back up. The very first classification system was by Aristotle, which you might have heard of. He was a philosopher, a Greek philosopher, and he was the first person to really start to classify organisms. He had 2 kingdoms, plants and animals. Back then, they didn't have microscopes, so they didn't know about bacteria or protists. They could see fungi, but they would have put fungi probably with the plant kingdom because if most people, if they look at a mushroom, they're going to say that looks more like a plant than an animal. So once microscopes got invented, scientists could look at cells. We could see much smaller things and be able to put them into more categories. So eventually, they had 5 kingdoms. This is what we had when I was your age. 5 kingdoms, which was Animalia, Plantae, Fungi, Protista, and Monera. Monera is bacteria kingdom. So in 1990, we have this guy called Carl Woese. He was studying bacteria, and he was looking at the molecular structure of their RNA and looking at their gene sequences in their RNA. And he also discovered that some bacteria have a certain chemical in their cell wall and some do not. Some bacteria like to live in extreme conditions and some do not. So he realized that bacteria, you can't just group them all together because they're very, very different types of bacteria. So we ended up about that time with a new kingdom, or actually 2 new kingdoms. They split the Monera kingdom into 2. So we end up with 6 kingdoms after that. The first kingdom was called Archaebacteria, which are the ones that prefer to live in extreme conditions. And then what you would call the regular bacteria are in the kingdom Eubacteria. So the 6 kingdoms that we have Archaebacteria, which is the newest kingdom. These are kind of like the weird cousins of the bacteria that we're familiar with. Like I said, they live in extreme conditions, so places that are super hot, super cold, super salty, down in those hydrothermal vents under the oceans that I showed you a picture of the other day. So they're like the weird cousins that go off and hang out in weird places. The Eubacteria, which was previously the Monera kingdom, these are your regular bacteria, the one you come into contact with every day, staph and strep, things like that. Then you have your Protista, which is kind of like the odds and ends kingdom. Protista share characteristics with 3 of the other kingdoms, but they're not quite able to be placed in those kingdoms because they have some different characteristics. So you have algae, which a lot of people would think would be in the plant kingdom, but plants have to be multicelled and algae are single-celled. So they're going to go into the kingdom Protista. You have protozoa, which are like little animals, but again, they're single-celled, so you can't be an animal if you're single-celled, so they go into Protista. And then your slime molds are similar to a fungus, but not quite. So I like to say Protista is the odds and ends kingdom that doesn't really fit into anything else, and they're all very different from each other. So I predict someday we're going to have the Protista kingdom breaking up into 3 separate kingdoms. Then you have your fungi, which is molds, mushrooms, yeast, mildew, things that you're familiar with that you encounter occasionally. You have your plants, you know what plants are, and then you have your animals. So Woese came up with this tree of life system where he basically tried to make a family tree similar to what Darwin attempted a long time ago. He added another taxon above kingdom, which he called domain. And there are 3 domains, archaea, which is the archaebacteria, bacteria, which is the eubacteria, and then eukarya, which is the eukaryotes. So anything that has a nucleus is in the same domain, which is over here. So animals, plants, fungi, and protists are going to be over here. And then each type of bacteria has their own kingdom because they are so not closely related to anything else. They're so far away on the tree of life from everything else. So this table's in your book. This just goes through some of the characteristics that these kingdoms have, and you can see why they're put in certain kingdoms that they are. So for unicellular, all bacteria are unicellular. Protists and fungi have some of each, and then all plants and animals are multicellular. We said these 4 that are in eukarya have a nucleus. Bacteria do not. There's differences in the cell wall. Remember, animal cells do not have a cell wall. Everything else has the possibility of having a cell wall, and their cell walls are made of slightly different things. Making their own food. Some kingdoms have both autotrophs and heterotrophs. The only ones that are for sure, fungi have no autotrophs, animals have no autotrophs, and then plants have no heterotrophs. And then it talks about reproduction a little bit. We'll get into that more when we study reproduction. So looking at Woese's system, he came up with this idea of what are called phylogenetic trees, which are the tree of life that we've seen before, and it just shows the relationships between different living things. So it's just like a family tree. You have a common ancestor, which is going to be at the base. Here's the common ancestor, and then everything kind of branches off from there. We talked about the formation of new species being called speciation. So each time speciation occurs, those 2 new species share a common ancestor. So the study of all this is called phylogeny, which is the study of evolutionary relationships. And this is how phylogeny is represented with this tree of life. And you'll see different ones. This was Woese's original tree of life from the 90s. This is one that your book made. It's very, very similar, but they've separated out the 4 groups of eukaryotes. And you can see, like I said, within Protista, there's lots of different groups within Protista. So they'll probably break apart and add some more branches to that tree of life eventually. So phylogenetic trees, you might see one of these on a test. They can look like a tree, okay, like this, or you might see them looking like brackets, like a basketball tournament or something like that. But they're giving the same information. They're saying that this organism is the common ancestor of these organisms, and just wherever there's like a dot or a branching off in the bracket, that's where they split apart. So like I said, common ancestors are represented every time there's a split or a branch in the tree. Okay, these little dots are called nodes, and those are just representing the common ancestors. Okay, and they're also representing a time when speciation occurred in history. So evolutionary time is going to be progressing from the trunk to the branches to the tips. So this number 1 here is our oldest organism, and then this one is very helpful because they're numbered. But if you look at the numbers, those are the numbers that are showing time moving forward. So organism F and organism E are going to be the farthest from the original point. So those are going to be the organisms that came about last. They also try to kind of indicate that with the length of each branch. So a shorter segment is going to represent a shorter period of time than a longer segment. So the organisms labeled at the tips of the branches, these organisms, are going to represent organisms that are known. They can either exist today or they might be extinct organisms. If you have a more recent common ancestor, you are more closely related. So we have organism F and organism E. They're fairly closely related, and they would be more closely related than, say, organism F and organism A. So it says, study the figure below. Which statement is true? We have A, organisms C and D are more closely related than organisms E and F. Okay, so here's E and F, here's C and D. We have B, organisms C and D evolved at the same time. Okay, and they're very, very close, but notice they're not sharing that dot. Okay, so that's not going to be true, and that eliminates A and B. We have C, organisms A and F lack a common ancestor, okay, which is not true. You know, they're pretty far apart on here, but they still have this common ancestor down here at the trunk of the tree. And then we have D, organism A is an older species than organism F, okay, and we know from what I've said previously, that's going to be the one that's true, okay. Organism A branched off from the tree before organism F did. So another version of this is called a cladogram. Okay, and a cladogram focuses more on specific characteristics that organisms share and what actually makes them related. So when you look at this cladogram of all of these vertebrates, these are all vertebrates. So you have some fish here, you have an amphibian, a reptile, a bird, and then 2 mammals. You notice down here you have the characteristics that put them into groups. So right here where you have jaws, okay, a hagfish is a jawless fish. A hagfish does not have jaws. It has like a raspy mouth that sucks onto stuff, whereas a perch has a jaw that opens and closes. Okay, the difference between a fish and an amphibian is going to be lungs. Fish have gills, amphibians have lungs. Okay, a difference between an amphibian and a reptile is claws or nails. And then you go up here to feathers for birds. And then the thing that makes you a mammal is having fur and having mammary glands to produce milk. So the cladogram analyzes adaptations or characteristics that have evolved over time, and they call these things derived characteristics. So you're going to need to be able to interpret a cladogram. And evolutionary time is, we're going to start at the bottom again, like we did with the phylogenetic tree. So these are going to be the things that evolved earliest. And this is a similar example. We have our jawless fish again. We have our lungs. This time we have lays eggs is going to be the difference, and it has to do with the type of eggs. Fish have the squishy eggs, whereas a snake and a crocodile have the leathery eggs because they're a reptile. And then the difference between a reptile and a bird is going to be feathers. Okay, so these are going to be the characteristics that evolved first. These are going to be the characteristics that evolved last. So derived characters are labeled on the cladogram. So any organism that's after each of the characteristic is going to have that characteristic, but if any organism that's before that is not going to have it. So if you look at feathers, a bird is after feathers, meaning the bird has feathers. None of these other things have feathers. So it says, which organisms represented by the cladogram below have lungs? So here's lungs. Anything after that point is going to have lungs. So the snake has lungs, crocodile has lungs, the bird has lungs. All right, study the cladogram below and see if you can answer the following questions. It says, which derived characters do mammals share with crocodiles? Okay, so here's mammals, here's crocodiles. Okay, it says crocodiles and mammals share all the characteristics that appear to the left of crocodiles. They both have 4 limbs. Okay, they both have amniotic eggs. Okay, here's 4 limbs, amniotic eggs, and additional skull openings. Okay, so again, anything to the left means they have it. It says, which derived character makes mammals unique? That's going to be fur. Which derived character do crocodiles, birds, and snakes share, but not mammals? Okay, that's up here. So you got to kind of look around and be careful there. So these are all grouped together in eggs with shells. Okay, we do not have eggs with shells. And it says, even though derived characters are not labeled that make birds and snakes unique, which characteristics could you identify? Well, this is based on your knowledge that birds have feathers, and something that makes snakes unique, no legs, possibly. And it says down there, remember that a loss of a characteristic can also be a characteristic. Like if you don't have something, that's a characteristic. So that would be good to use for snakes. All right, here's another one. The table below gives derived characters, either present or absent, in 5 different organisms. How could these species be presented in a cladogram? So using the chart, determine the number of derived characters present in each organism. You're going to identify the organism with the fewest derived characters. That's going to be the one to the far left. In this case, it's the bacterium. It doesn't have any of those things. So you're going to put your first branch for the bacterium. Then you're going to look at the one that has the next least. Sponge only has one thing, which is multicellular. Add that on. Okay, and then you're just going to keep repeating your steps, and that's how you make a cladogram. On a test, obviously in class, you're not going to have to build anything because it's going to be multiple choice. On the state test, they'll definitely have you look at a cladogram and interpret a cladogram. I don't know if they'll make you build one, but they could because they have that drag and drop option thing. So they could potentially have you build a cladogram. So just remember, all you have to do is look for which has the least amount of characteristics. That goes on the left, and then just go from there. And that is it for classification. As always, if you have any questions, please let me know.